Hi everyone, welcome to CAPSA. Today's topic is about microscopy. So microscope, a good microscope should have three properties. First one is good resolution, second one is good contrast, third one is good magnification. Resolution is a power which refers to the ability to produce separate images of closely placed subjects so that they can be distinguished as two separate entities. Okay. And good contrast can be improved by staining the specimen. Good magnification can be achieved by using concave lenses. So resolution depends on refractive index. Oil has higher refractive index than air. Okay. And good contrast can be achieved by staining the specimen. And magnification can be achieved by using concave lenses. Okay, this is the refractive index of human eye light microscope and electron microscope. This is the mathematical formula of refractive index. You can see here, I am not repeating. So what type of lights used in microscope? Three type of mainly light used in microscope. First one is transmitted light, second one is reflected light, and third one is polarized light. So transmitted light actually used in bright field microscope and reflected light used in dark field microscope and polarized light used in differential interface contrast microscope. Okay, these are the mathematical formula of numerical aperture, which is equal to n sin alpha, n equal to refractive index or Ri, alpha equal to 1/2 angular aperture of the light. Resolving power equal to 0.61 into wavelength by Na or numerical aperture, or 0.61 into wavelength denoted by lambda by Na or n sin alpha. Now what are the type of microscope? Bright field microscope forms dark image against a brighter background, and gas vacuoles, nuclear materials, cytoplasm, pigments, etc. are studied under this kind of microscope. In dark field microscope, the object appears bright against dark background, and this can be so much made easy by using a special kind of dark field condenser and spirochetes kind of unstained cells. These are actually identified by this kind of microscope. In phase contrast microscope, specimen can be visualized by creating difference in contrast between the cells and the water, and it converts slight differences in the refractive index and cell density into easily detectable variations in light intensity. And this kind of microscope used to study microbial motility, determining the shape of living cells, detecting bacterial components such as endospores, inclusion bodies, etc. In fluorescence microscope, when the fluorescent dyes are exposed to UV rays, they become excited and are said to be fluorescence. This kind of phenomenon used in this kind of microscope. They actually converts these invisible short wavelength rays into light of the longer wavelengths or visible light. Okay, and under this microscope, malarial parasite, cyclospora, Mycobacterium tuberculosis are observed under this kind of microscope. Electron microscope actually invented by Ernst Ruska in 1931. Okay, so it is invented by Ernst Ruska in 1931. Okay, and it is of two types. And in this microscope, specimen actually visualizing by using beam of electrons, which produces magnified images. Diffraction of light is used in this type of microscope. Okay, electron microscope. So electron microscope are of two types, TEM and SEM. TEM is transmission electron microscope, SEM is scanning electron microscope. It has a resolution power 0.5 nanometer. It has resolution power of 7 nanometer. Okay. It shows always 2D view. It shows the 3D view. Okay. And internal structure are studied under this microscope, and in SEM actually surface structures are studied. Okay. So this is bright field microscope, and the image created under bright field microscope will looks like this. This is the dark field microscope. Image is this. This is phase contrast microscope, and image will look like this. This is fluorescent microscope. Fluorescent dyes are used here. The image will look like this. This is electron microscope, and this is the surface view of red blood cells. You can see same image, scanning electron microscopy. Okay. So this is the end of my lecture on microscopy. If you enjoy my lecture, please do like, comment, share and subscribe. Thank you for your attention.